Good morning, Professor and classmates. I'm Song Young-jong. I study in Professor Kim Jae-yeon's lab about plant genome engineering. Today, I want to talk about what I learned in the lab. First, I will talk about necessity of CRISPR-Cas system in plant genome engineering. After that, what I learned in the lab. And then I will talk about competitiveness of CRISPR system-based plant breeding technology. Coming to the necessity. Although the world population continues to increase and the population growth rate has declined since the 1950s, from the estimated 7.7 billion people worldwide in 2019, and the UN and FAO are projected to increase to 9.7 billion by 2050. In order to be responsible for the food of this growing population, 70% more food needs to be secured than present. But as urbanization progresses, even cropland is decreasing. Therefore, we need to increasing crop productivity. Traditional breeding methods include isolated breeding to select natural mutations, cross breeding with allied species or natural species, and mutation breeding using chemical or radiation. But they all have disadvantages of long time and labor. Above all, these are a weakness that the pool of available allele is limited. As a method of overcoming the limitation of conventional breeding, GM technology was introduced to introduce a new external gene, and GM breeding showed rapid growth based on its innovativeness in the last 20 years, and has succeeded in commercialization. However, it's still distrusted by consumer and environmentalists, so its growth is limited by strict regulation. This leads to a distorted structure in which only multinational corporations entered the GMO market. For this reason, our lab wants to get the elite line relatively and safely with CRISPR. Coming to the next, what I learned in our lab. Now, I am studying about CRISPR. Then, what is the CRISPR? The CRISPR-Cas system is a prokaryotic immune system that confers resistance to foreign genetic elements such as those present within plasmids and phage that provide a form of acquired immunity. RNA harboring the spatial sequence helps Cas protein recognize and cut foreign pathogenic DNA. Other RNA-guided Cas proteins cut foreign RNA. CRISPR are found in approximately 50% of sequenced bacterial genomes and nearly 90% of sequenced archaea. Since then, many people have preceded it, and single guide RNA linked with cRNA and tracer RNA. Cas9 protein with cleavage activity was expressed together to enable site-specific cleavage. I applied this CRISPR system to plant genome engineering, so I'm going to explain how this system is used for plant genome engineering. First, select the target gene such as biotic or abiotic stress resistance gene, target gene for crop productivity improvement. And then design the guide RNA. When you design the guide RNA, if you want to completely remove the function of the gene, it's better to cut the front exon. And if you want to reduce the function of the gene, it's good to cut the rear exon. And the palm site required by each Cas protein must exist. It should be designed according to the optimal guide RNA length of each Cas protein. After selecting the target gene and designing the guide RNA, cutting the target gene, insertion or deletion mutation occurs in the plant by the DNA repair system. Then, if the expression of the gene decreases or disappears due to the indel mutation, a plant having the desired trait can be obtained. After that, homozygote can be obtained using self-pollination around 1 to 2 generations. So, how to introduce single guide RNA and Cas protein complexes into plants? In our lab, use agrobacterium-mediated plant transformation method. The following pictures show how agrobacterium infects plants. First, detected a chemical gradient of a ligand and activation of virulence. Second, tDNA excision. Third, movement of tDNA out of the agrobacterium. Fourth, nuclear import and integration of tDNA. Fifth, expression of tDNA and pathogenics. Our lab used the following this plant transformation method. Agrobacterium having a plasmid capable of simultaneously expressing Cas9 and single guide RNA is co-cultured with X plant. Then the plant into which the tDNA is inserted grows in the selection medium. However, plants without tDNA inserted are bleaching and died like the white plants showed in the picture on the right. The selected plants are transferred to soil and grown. Next, how to analyze plant. First, collect the tissue of the plant grown in the soil. Then isolate genomic DNA. After that, the target region is amplified by PCR to confirm mutation. This is the result of single sequencing of isolated DNA and analysis of using genome engineering analysis software. From the single sequencing result, we can see that the double peak is partially visible. As a result of analyzing with software, it can be seen the 3 allele types appeared. 1 wild type and 2 deletion alleles are observed. Self-crossing will yield homozygote traits. Coming to the last, what is the competitiveness of our lab and for me? Currently, our lab is trying to make a plant with resistance to biotic or abiotic stress. This could quickly gain an elite line and contribute to securing global food security. Thank you for your attention.